Hello, everyone, and welcome to today's broadcast, Automated Variant Classifications in Qiagen Clinical Insight Shows Near Perfect Coordinates with ClinVar Expert Panel Assessments, presented by Dr. Jennifer Poitras, a Principal Genome Scientist at Qiagen. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Qiagen. For more information about our sponsor, visit them at www.qiagen.com. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type those questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, just click on the Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credit tab located on the top right corner of your presentation window and follow the process to obtain your credits. Without further ado, please join me in welcoming our presenter, Dr. Jennifer Poitras. I will now turn the presentation over to her. Welcome, Dr. Poitras.
Thank you for the introduction and for the opportunity to speak with you all today. So today I'll be talking about Qiagen's clinical decision support tool, the knowledge base underlying it, and focusing on how we leverage that information to automate ACMG variant classifications, and more specifically, how these classifications show near perfect concordance with ClinVar expert panel assessments. So in the way of a legal disclaimer, the tool I'm discussing today, as I said, is a clinical decision support tool and isn't intended for diagnosis, and this message is meant to convey that. So for today's agenda, first I'll go through an introduction to the Qiagen knowledge base, then introduce our Qiagen Clinical Insight Interpret tool and demonstrate how the knowledge base underlying it enables rapid and accurate variant interpretation. We'll also see how automated variant classification is utilized by invoking all of the ACMG rules to align with the guidelines and some benchmarks that show the value of the tool. So today, we're really focusing, as you can see at the end of the pipeline with the red box around it, or the tertiary analysis or interpretation. But just to put things in context, the goal of Qiagen is to take you from sample to insight, where we offer a variety of DNA and RNA extraction kits, as well as library preparation kits to prepare you to perform your next generation sequencing. Once you've generated your FASTQ files, you can easily feed those into the Biomedical Genomics Workbench, which is our graphical user interface application. That's truly a one-stop shop for NGS analysis, empowering the scientist or clinician with limited bioinformatics knowledge to map the reference and, for the purposes of today's discussion, call variants to produce a VCF file. Which brings me to the topics that I'll cover today and how we can leverage our curated content within our downstream tools, particularly Qiagen Clinical Insight, which is our clinical decision support platform, to rapidly distill the list of variants down to one or a handful that are likely implicated in the patient's disease. So the end result or output from QCI will be a report with relevant variant information, including automatically computed ACMG pathogenicity, which you can see on the left, as this EP300 variant is pathogenic. And also this including citations in the second page that the user has added that we've curated within the knowledge base. And this is really facilitated through having all the content listed here within the knowledge base, and that's really the focus of today's discussion. So today we'll be mainly talking about hereditary disease indications, but I just wanted to point out that this is a platform agnostic NGS interpretation tool, meaning you can perform your sequencing on any platform you like and just take the VCF file. And for the somatic indications, we use our curated content to tier the variants by the AMP tiering classification system in terms of actionability, and also using the ACMG guidelines for both hereditary panels and also clinical exome indications. Which brings me as a nice segue to my next slide. So just as an introduction to the ACMG guidelines, for those in the audience that may not know this system, several years ago, the ACMG, or American College of Medical Genetics, created this evidence-based framework for guidelines of variant interpretation. So rules falling into various evidence categories that favor a variant as benign or pathogenic along this continuum, from left to right. With also different strength categories you can see listed at the top of each column. So what the variant scientists will do is they'll look to see, which of these criteria does my variant meet, and those criteria in aggregate classify a variant as benign, likely benign, a variant of uncertain significance, likely pathogenic, or pathogenic, depending on the combination of rules applied. And you can read more about this framework in the publication down to the right. So identifying which rules to apply to interpret variants is no small undertaking. So many resources are necessary for gathering that information, and I've just highlighted a handful here. So the variant scientist would need to sift through variant-specific publications from PubMed, check public and proprietary databases to see, if my candidate gene or variant has been seen before in my disease context, and also looking at in silico predictors as well as public databases to look at allele frequency. So what Qiagen has really done is simplified this process by putting it all in one place, but there are two major challenges that a variant scientist faces when having to sift through all this information. One is of efficiency, because this is very laborious and time consuming, and the second is of accuracy, because a person might miss something having to look in all these different places. So what Qiagen's done is simplified this process by centralizing it and putting it all in one location that we know as our knowledge base. So as I said, we put all this information and aggregated it into what's known as the Ingenuity, or really Qiagen knowledge base. So for the past 20 or so years, we've had MD and PhD level scientists manually curating the literature and inputting findings in a controlled and meticulous way. Beyond the literature, we also incorporate data from third-party sources like ClinVar, OMIM, and clinicaltrials.gov to come together and build this ontology such that we have a rich resource of gene-gene and gene-phenotype relationships that you'll see a little later in the presentation will enable us to easily pull out variants of interest from a clinical exome. Additionally, these findings are what are tied to those ACMG rules that invoke them during the variant interpretation process. So we've talked a lot conceptually about our content, but let's look at the breadth of coverage in terms of numbers. So our knowledge base not only contains two of the most widely used databases of clinically relevant variants, as you can see HGMD in the gray circle, and genes in ClinVar in the sort of periwinkle circle. But you can appreciate how much value is added from our unique curated content from the literature. As on the left, you're seeing that we're adding 121% more genes on top of what's already contained within HGMD and ClinVar. And on the right, you can see that we're adding 159% more variants just from curating from the literature, including or on top of what's contained in HGMD and ClinVar. So you see the value that our manually curated content has added. And just to show you how this information is presented, here's an example of how these variant-specific publications are curated into findings. So there are 3 findings associated with this publication where we can see this variant is associated with a particular disease, EP300, is a variant in the EP300 gene is associated with Rubinstein-Taybi syndrome, and within a particular population, as you can see the El Salvadorian population down below in the last finding. So ultimately, it's these findings that are used to invoke the ACMG rules and compute pathogenicity. So now that I've introduced the Qiagen knowledge base and how the content is used to automatically classify variants, let's look at some benchmarks that show how QCI addresses the major challenges faced in variant interpretation. So I mentioned in the beginning that there are 2 main challenges faced by the variant scientists or geneticists in mining and utilizing all available information to classify variants. So now I'll touch on a couple of benchmarking studies that demonstrate how Qiagen Clinical Insight addresses these challenges of efficiency and accuracy. So first, we'll start with efficiency. So this is a study performed by Counsel, and Counsel is a health technology company that offers carrier screening and hereditary cancer risk assessment. So at last year's American Society of Human Genetics meeting, Counsel presented their findings in a poster from an evaluation they performed, and the goal of this evaluation was to assess whether utilization of Qiagen's variant-specific bibliographies within QCI could match the level of accuracy and quality of Counsel's more time-intensive manual article selection approach. They looked at around 2,000 variants they reviewed over a 3-month period, and in the graph on the left, you can appreciate the comprehensive nature of our content, where there's a large amount of overlap between our variant-specific citations and those present in the Counsel House database, and there is a huge amount of, in the center bar you can see there's a huge amount of unique information in the Qiagen knowledge base alone. And not only does QCI provide a comprehensive content resource, but this centralized content reduced the time spent searching for variant-specific publications significantly. And this is made most obvious when looking at the median, where the search time is reduced in the graph on the right by an order of magnitude when using QCI, and these are represented by the yellow bars. So overall, Counsel demonstrated that there was a clear value in adopting QCI, and they've now made it a key component of their variant assessment pipeline. So now we've addressed efficiency. Well, the other challenge we mentioned, right, is accuracy. So I mentioned that we are uniquely positioned because of our curated content to invoke all 28 ACMG rules, but how accurately are we in doing so? So to test this, we took expert panel assessed BRCA1 and BRCA2 variants from ClinVar, which you can see in the top window, and put those through QCI to compare the Enigma assessments with the classifications in QCI. So I should mention that Enigma is the expert panel that is extremely dedicated to determining the clinical significance of BRCA1 and 2 variants, as well as other known or suspected hereditary breast cancer predisposition genes. So in this study that I'll be speaking about today, there were over, at the time the study was performed, just over 6,000 of these variants. And in this particular example, you can see that this is an example of a concordant variant, where the variant was classified as pathogenic in QCI and was also called pathogenic by the expert panel. And what we found was actually, rather astounding. So in this graph, the green bar represents classifications made by Qiagen or QCI. The green bar represents those made by the expert panel. So you can see that and you can see at the top of the graph it says N equals 6,135, as those are the number of variants that we benchmarked here. That 81.7% of those were perfectly concordant between QCI and the Enigma expert panel. And then when we move to the right, you can see that an additional 17.87% were different, but those differences should not affect medical management. As you can see, for example, likely benign versus benign, which shouldn't affect medical management of the patient. So when we take those two together, we can see at the top where I've written 99.6%, there's 99.6% concordance with respect to clinical actionability when we take these together, which is, rather incredible for, a computer algorithm. So now we can also look at on the right, there is this very small percentage, so 0.43% of differences that might affect medical management. So those are the key sources of those discrepancies fall into a couple of different categories. So one is the Enigma-specific variant scoring criteria outside the classic ACMG framework. So for example, there are pieces of information or data like posterior probability, for example, that are not part of the ACMG framework that Enigma does use. So we don't have, the algorithm doesn't have control over that because they're using information outside the framework. Additionally, there are some functional studies that were not considered by Enigma, and we'll go through an example where we highlight that in a second. So first, let's just go through an example of a concordant variant. So here you can see this particular BRCA2 variant was considered pathogenic or classified as pathogenic in QCI and also pathogenic by the ClinVar group. And I just wanted to point out, exactly what this looks like within QCI. So you can see that we show you which rules, and you can see the criteria ID of PBS1, for example, and PS3 going down. So we show you, which criteria were actually invoked. And then additionally, to the right in the evidence column, we can show you, how many publications actually are underlying that particular rule that invoked it. And you can go and investigate those. And we'll do that now. So we look here and we can see that PS4 was triggered. Now this is actually not trivial to do because this rule is looking at, is the prevalence of this variant enriched or statistically significant in the disease population over controls. So to do that, you need clinical cases, which we've curated from the literature, and then perform a Fisher's exact test. And you can see in this red box that we actually have done all of that and provide you with the p-value. And since the p-value is significant, we invoke PS4. You'll also notice in the evidence column that there are 24 publications that were used to collect those individuals. And you can actually go in and investigate, those 24 publications to see which individuals, how many individuals were in each of those publications. Is it a family study or an individual? So we keep everything extremely transparent within QCI to provide you with all of the evidence that was necessary in coming to this assessment. So now we'll look at a discordant variant, okay? So for this particular synonymous variant, the Enigma panel called this likely benign, and you can see in the evidence that they're citing an in silico prediction, and that's the evidence that they're providing. However, in QCI, we actually call this variant likely pathogenic, and this is using, of course, the standard ACMG framework, and you can see in the evidence column I've circled the two because this is there are two curated functional studies that indicate this variant results in a splicing defect. So we've actually curated functional studies that were not considered by Enigma. And this is actually a good point to bring up right here is that the Enigma doesn't actually consider functional studies currently because there's still, within the group, they're debating, what kinds of functional studies actually merit inclusion in variant scoring for BRCA1 and 2. So what we're doing is we're just providing you with all of the possible information so you can make the judgment call of what functional studies you'd like to include. So we've curated these two functional studies that are then pushing this into the likely pathogenic category. And when I investigated them, these publications actually do say that this particular variant leads to a defect in splicing in a mini-gene assay when I went through and looked at it. So it shows you the power and the importance of our curated content in kind of evaluating the pathogenicity of a variant. So in summary of these benchmark studies, so the curated bibliography from the council study reduced time spent searching for variant-specific publications by an order of magnitude. So that addresses the efficiency issue that's faced by the variant scientist. Also, in the Enigma study, QCI automated ACMG classification of over 6,000 BRCA1 and 2 variants, resulted in 99.6% concordance with the Enigma expert panel variant assessments. With respect to clinical actionability. And the sources of this discordance, as I said, were due to kind of nuances in the Enigma scoring framework. And finally, as always, we're continuing to refine the ACMG classification algorithm in concordance with the sequence variant interpretation working group's recommendations to ensure the highest level of accuracy and alignment with ACMG standards. So now we've seen how automated ACMG classification works and proven its accuracy, but let's look at a practical example where we identify a causal variant from a clinical exome sample. So just briefly to discuss the clinical exome framework or workflow. So in this particular example, you can see in the pedigree on the right, we've got two unaffected parents and an affected son. And this patient has a constellation of features, but no clear diagnosis, which is typically the case for a clinical exome. So you can see that the pedigree is shaded with these 5 different phenotypes. And all of this clinical information is put into a simple JSON file. In this case, phenotypes was used to contain all of the clinical data necessary from all of these patients and these family members and the relationships within it. On top of that, in addition to that JSON file with the phenotype data, we bring in the VCF files. In this case, we have 3 VCF files, 1 for each individual, but we do support, beyond trios, also more complex family structures as well. So by bringing the clinical data as well as the variant data into QCI, we can then use information in the knowledge base to filter that down, including allele frequencies, predicted deleteriousness, is the variant in the proband or not, to distill that down to a more reasonable list, and finally arrive at our variant interpretation. So we'll be using a phenotype-driven ranking algorithm to rank these variants, where you can see we've added those 5 phenotypes that were in the JSON file in the proband, and those 5 phenotypes appear as little rectangles going across. So all 5 phenotypes are overlapping with Rubinstein-Taybi syndrome type 1 based on curated information in the knowledge base. And additionally, EP300 is known to associate with Rubinstein-Taybi syndrome type 1. So you can see how EP300, this variant in EP300, is ranked at the top based on our phenotype-driven ranking. So to just go through this a little more deeply. So first, you can see I've drawn a box on the right around this intellectual disability and ACMG 59 view. So here we've created some virtual panels to subset the genes in the analysis. So in this case, the referring physician was concerned that maybe in this defined list of about 2,000 genes associated with intellectual disability, the patient has a variant in one of those genes, so they wanted to see and look at those genes separately, as well as the ACMG 59 genes, to see if there were any incidental findings. So in this particular case, you can see this red, the EP300 gene at the top, which ends up being our causal variant, and I'll show you that in the next slide. But that ends up being present within the intellectual disability subview. So you do have the ability to set up your own virtual panels as well. But as far as the phenotype-driven ranking algorithm goes, here again, you can see that these gray boxes in the third column over where it says candidate disease, so those 5 rectangles each represent one of the phenotypes that's displayed in the patient. And you can see in the first row that all 5 of them are present and consistent with Rubinstein-Taybi syndrome, which those connections are in the curated content in the knowledge base. Additionally, EP300 is known to be associated with Rubinstein-Taybi syndrome, and for this reason, that pathogenic variant, which is also automatically computed and classified as pathogenic, is our #1 ranked variant. You can look at other information in the table to kind of distill down or bolster this analysis. So for example, I can see in the mode of inheritance column that this is a dominant disorder. And then when I look at the genotypes of the parents in the proband, this is again consistent. So here in the proband column, you can see that this EP300 variant is heterozygous and present in the proband, but is absent in the unaffected parents, which again makes sense. Furthermore, stating or showing us that this pathogenic EP300 variant is causal for this disorder. To dig in a little further, we can go to the variant details tab, and this is where we're shown, as I showed before, which criteria were actually invoked, which ACMG rules were invoked, and I can review the evidence there as well as looking at the bibliography where I've drawn this arrow. So this will show me all the curated content. I no longer have to go to PubMed to search for EP300 specific publications, and I can add those to the report as shown here. So there are 20 references at both the gene and variant level that associate EP300 with a particular disorder that I can investigate or choose to add to my report. So all in all, the Qiagen knowledge base within QCI facilitates a number of things and is extremely powerful because of all of the curated content. So we've just shown how it can be used to rapidly filter and also prioritize variants from a clinical exome. Also, we use this curated content to uniquely position us to automate ACMG classification, as I mentioned, driven by this extensively curated content. We showed in 2 different benchmarking studies that this curated content increases efficiency and turnaround time in reporting variants, as shown by the council study. And then finally, yes, we can automatically compute ACMG classifications, but we also do so in an extremely accurate manner. that we've shown is highly concordant with the BRCA1 and 2 variant assessments of the ClinGen expert panel group, or ClinVar. So with that, thank you for your time, and I'm happy to take any questions that you may have.
Thank you, Dr. Poitras, for that informative presentation. And we will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appear on the screen, and click that Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question, Dr. Poitras, is how frequently is the knowledge base updated?
Okay, and that's a great question. So as I mentioned, we have several sources of content. So the content in the literature is actually updated basically daily, or we like to say on a weekly basis, so we're constantly updating that. Anything that we're getting from a database such as ClinVar and HGMD or OMIM, for example, those are all updated on the quarterly basis.
Thank you for that. We have one more question coming in. Does the user have the ability to change the ACMG rule that are applied?
Yes, in fact, they do. So as I showed you, I'll go back a couple of slides. So you can see that there are criteria listed here, and you can see the criteria IDs. So there are little X's next to them where you could delete those criteria, or underneath you can see the Add Criterion button. Additionally, you can change the strengths of those rules as well. So there's a lot of plasticity and sovereignty that we give the user in terms of amending those ACMG rules that are applied.
Dr. Poitras, thank you for that informative presentation again and for your important research. I'd also like to thank LabRoots and our sponsor, Qiagen, for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through February 2019. You'll receive an email from your LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thank you for joining us today. We hope to see you again soon. Bye-bye.